Okay, no problem. Let's start. So if good morning, good morning. If everyone quickly recall, in the last class, we have started discussing about the hypersensitivity, right? So we understand that there are four types of hypersensitivities are there. Okay. And we have discussed it till this particular slide. Okay. And once again, I'm reminding to all of you, please remember the hypersensitivities are basically caused by the what reasons? What we studied, friends, about the type 1 hypersensitivity, I told you that type 1 hypersensitivity is caused by what? IgE. But what is the statement we have used, friends? It is just not caused by the releasing of IgE, but what we studied, it is caused by the elevated levels of IgE. This is the one which will cause the type 1 hypersensitivity. And if you look into the type 2 hypersensitivity, what you have to understand, I told you, right? Antibody mediated. It is not a specific type of antibody because we discussed, right? Here, antibody is going to activate the complement system also. At the same time, antibody is going to activate the cytotoxic T cell also. So in a simple words, whatever the activity is shown, that activity shown by the help of antibody. That's why we can call it as the antibody mediated. And friends, if you look into the type 3 hypersensitivity, what you understand, it is mediated by the complex, which means there should be a presence of the antigen and there should be a presence of the antibody as well. If you recall type 4, what we discussed, friends, in the type 4 hypersensitivity, there will be an activation of many groups of cells. That's the reason we will be calling it as the delayed type of hypersensitivity or DTH, right? So in the last class, we have discussed it till this particular slide. Even I have explained you this slide. Now let's start one by one briefly. Let's start with the first one, type 1 immediate hypersensitivity. Okay, if everyone quickly recall what I have told you about the type 1 elevated level of IgE. Friends, among all the types, I can say that type 1 and type 2 are the most important for the exam point of view. Whenever they ask the question, mostly they can ask from either 1 or 2. So make sure that you'll understand the concept very correctly. Follow me, everyone. Allergens and atopic individual. Friends, listen my statements very correctly. What are allergens? Allergens are the one which basically cause the allergic reactions. A simple words. Allergens are what? See, what is the line I have written? Try to understand what I have written here. They are non-parasitic antigens that are capable of stimulating the type 1 hypersensitivity. Remember, friends, why I am using the word non-parasitic antigens? Because they are not at all taking shelter in the host body. Let's take an example, pollen grain. I told you many times, whenever we go to a green park, some people, what happen? All of a sudden, they start sneezing, their eyes become completely reddish, running nose, difficulty in breathing. These all will occur. Why, friends? Here, whatever the pollen grains that are present in the air, remember, they are acting as what, friends? Allergens. They will stimulate the mast cell. Mast cell release the histamine. Histamine will cause the constriction of the bronchi, so which leads to the difficulty in the breathing. So what is the point you have to remember, friends? Allergens are generally non-parasitic antigens, which are having the ability or capability to stimulating the hypersensitivity response. Okay, now listen to me carefully. What are atopic individuals? Friends, so the same example. Let's take in the same example. Suppose example, 3 people visited the park. When 3 people visited the park, only one person facing all these difficulties like difficulty in breathing, running nose, eyes becoming completely reddish. Not everyone. So what is the point you people has to understand? Those kind of individuals who are sensitive, in other words, you can call it susceptible, who are sensitive to the allergies, who are susceptible to allergies, those kind of individuals called as atopic. In other words, all of you listen my statements. How can I define the word atopic individuals? Follow me, friends. Atopic individuals are those type of individuals who is having the genetic tendency. What, friends? Who is having the genetic tendency to show the susceptibility or to show the sensitivity to the allergies. Those kind of individuals only, friends, we will be calling it as the atopic individuals. Don't get confused. I am not using the word in the atopic individuals. I am not using the word that they do not have the IgE. No, I'm not saying that. They do have the IgE antibody, friends, but what will be the level? The level of IgE will be extremely higher. Friends, you people keep it in your mind. We already studied that IgE is the antibody which is released in the serum at a very low concentration. So what is the point you have to remember? If the level of IgE becoming higher and higher, which indicates that there will be a allergy. So in the atopic individuals, what happened? The IgE level will be completely fluctuated. Most of the time, it will be releasing at the higher concentration, which indicates that these people can show the tendency towards the allergies. Don't get confused with that point. Okay, listen my statements very carefully, friends. Next 3 to 4 slides, whatever we are going to discuss, friends, they are extremely important. Please make sure that you understand the concept very correctly. Okay, just follow me correctly. Okay, look into this. Okay, look into this. This is the B cell and this is the T helper cell 2. Friends, I hope you all remember when we are studying the T cell development, we have studied it very correctly. What we studied, T helper cell 1, it is always associated with which cell? Antigen presenting cell, like macrophage or dendritic cell. I want you to understand the concept correctly. Then T helper cell releases what? Certain interleukins. Best example, interleukin 2. Now the interleukin 2 is activating what, friends? T helper cell 2. Now what the T helper cell 2 is doing, friends? Good morning, continue. What the T helper cell 2 is doing, the T helper cell 2 with the help of TCR receptor, MHC, CD4, it will activate the B cell. Once the B cell gets stimulated, we all know that, friends, B cell is going to differentiate it into plasma cell and memory cell. And friends, the plasma cell is going to synthesize the antibody. Once there will be a synthesis of the antibody, what is going to happen, friends? What kind of antibody it is? Everyone look into the picture. IgE, allergen specific IgE. Everyone follow me correctly. Now this particular IgE will come and attaches where? That's the question. Friends, on the surface of the mast cell, everyone remember my words. On the surface of the mast cell, there is an important receptor is present. We all know that. What is that receptor, friends? FC receptor. To that FC receptor, who will come and attaches, friends? To that FC receptor, immunoglobulin E will come and attaches. Simple point. The moment immunoglobulin E will come and attaches, what happen? Immunoglobulin E cross-linked with antigen or allergen. Once it cross-linked with the allergen or an antigen, what's going to happen, friends? Obviously, it is starting the process of degranulation. What is degranulation? Releasing the chemical. I told you many times, friends, what kind of chemicals are present in this particular mast cells or the basophils. There are many chemicals are present, friends. An example, heparin. What is heparin? Powerful anticoagulant. And there is a presence of a very important chemical, histamine, which is involved in constricting the bronchi. Correct, na? Even it contains a platelet activated factor, which is required for the platelet activation. And there is a presence of a very important chemical among all, which we can call as prostaglandins, which are responsible for stimulating the pain. So all these chemicals are present in the granules. When all these chemicals are going to release, friends, all these chemicals will release when the granules break. So now we are going to understand a mechanism, how exactly the granules are breaking. See, listen my statements correctly, don't get confused. I am going to explain to you how IgE will be involved in breaking down of all these granules and releasing all their contents. So listen to me very carefully, friends. If IgE wants to involve in breaking these granules, first of all, friends, IgE should associated with the receptors. Yes or no? Of course, yes. IgE should associated with the receptor. What is this receptor that we studied, friends? Fc receptor. Follow me correctly without confusion, friends. There are two types of Fc receptors are there. Follow me correctly. One we can call, friends, Fc epsilon R1 receptor, Fc epsilon R2 receptor. Actually, friends, listen my statements. Both the receptors can be used by the IgE. But most of the time, friends, IgE starts utilizing Fc epsilon R1 receptor only. What is the reason why it is using only Fc epsilon R1? Because, friends, IgE having the higher affinity towards this receptor. You people no need to remember the structure of the receptor, but I'm just saying this structure for your better understanding. Okay, look into the structure. Okay, everyone look into the structure. This Fc epsilon R1 receptor contain different domains. See, this is entirely receptor, friends. I'm just talking about this. This is the Fc receptor. I'm just talking about this exactly because IgE will associated with the Fc epsilon 1 receptor only. It contain alpha domain, it contain beta domain, and it contain the gamma domain. Friends, if you observe the structure very correctly, alpha, this is the alpha domain having a larger extracellular domain, correct now? And if you look into the gamma, gamma having what, friends? Larger intracellular domain, correct now? But if you look at the beta, it did not have a greater extracellular domain. At the same time, it do not have a greater intracellular domain as well. Just remember, only alpha having extracellular domain, very large I'm talking about, and gamma having intracellular domain. Even though beta having the intracellular domain, but it is not a larger domain. So these three together, they can form this particular Fc receptor. And keep it in your mind, Fc epsilon R2 cannot be used by the IgE because it is having the lower affinity. Because, friends, if the affinity is lower, IgE attachment will become lower. If the IgE attachment will become lower, obviously, friends, it never ever stimulate the process of degranulation. That's why all the time it will always prefers the high affinity receptor only. Look into the picture, everyone, without confusion. You can see, right? This is what cell? Mast cell. Mast cell contains what? Mast cell consists of an abundant amount of granules which contain various type of chemicals which are involved in the process of hypersensitivity. That is what we are studying. And this is the mast cell, and this is the Fc receptor. On the Fc receptor, who will come and attaches, friends? IgE will come and associated, or the IgE will come and cross-linked. Don't get confused here. Now I'm going to explain you. This is the most important slide you people has to remember. If you get the question, from the type 1 mediated hypersensitivity, there are many chances are there that you can expect the question from this particular slide specifically. Okay, follow me very correctly, friends. Listen to me very correctly without having confusion. See here, this is what? Fc epsilon R1 receptor. This is Fc epsilon R1 receptor. Don't confuse, friends. What is the ultimate goal? Please keep this in your mind. Ultimate goal is the degranulation. So how IgE is doing this process? Follow me correctly. Just listen to me. I'll explain twice. This is the IgE allergen Fc receptor. Once allergen cross-linked with the IgE. What happens, friends? Immediately IgE will stimulate protein tyrosine kinase, which is the PTK. Once PTK get activated, follow me everyone. PTK is going to activate an important effector molecule, which we all know very well. What, friends? PTK is going to activate an important effector molecule, which we can call, friends, a PLC, phospholipase C. Now, what the phospholipase C is doing, friends, we know that phospholipase C break down the membrane lipid. We already studied in the cell signaling topic. That is a PIP2. PIP2 into what, friends? A PIP2 into IP3 as well as the DAG. And we studied many times IP3 always bring the calcium. Now DAG and calcium together, they are going to activate the protein kinase C, which we can call PKC. This is fine. Now you have to remember a unique statement. What's the ultimate function of PKC? Anyway, what we have to do, we have to stimulate the process of the degranulation. So, you know, friends, what is going to happen now? This particular PKC, follow me, friends. Listen to me. This particular PKC will stimulate these granules. See, you only try to understand. This is the vesicle which contain abundant amount of granules. Follow me very correctly, friends. These particular granules has to come near the membrane. Understand what I'm talking correctly, friends. This is the granule which contains several chemical mediators. When the degranulation occur, friends, whenever this vesicle come and fuse the membrane, this is the membrane. Once they fuse the membrane, then the degranulation occur. Now the valid question is here, how this granule will come and fuse the membrane. You know, friends, what the PKC is doing. We studied in the cytoskeleton in the cell biology chapter very clearly that any movement that is happening inside the cell or inside the cytoplasm, everything is with the help of a cytoskeletal machinery such as the microtubule and microfilaments. Now, what's the unique point you have to remember is that this protein kinase C will come and stimulate the microtubules and microfilaments. So friends, whenever there is an activation of the microtubule and microfilaments, what happens? The granule starts moving because of the rearrangement. The granule starts moving towards the membrane. They will fuse it, then they will release the granules. They will break down the granules. I'll explain once again. Whomever did not understand, follow me correctly. This is the IgE. IgE will be associated with the Fc epsilon R1 receptor. Once the IgE cross-linked with the allergen, immediately the Fc epsilon R1 receptor will be active. Once it is active, it is going to activate the PTK. Once the protein tyrosine kinase get activated, it will further activate an important effector molecule such as phospholipase C or PLC. Now that PLC break down another membrane lipid called as PIP2 into two secondary messengers, DAG and IP3. What the IP3 is going to do? IP3 is going to bring the calcium out from the endoplasmic reticulum. Now the calcium and DAG together going to activate an important protein called as PKC. What the PKC is doing? It will cause the rearrangement of the microtubules and microfilaments, which leads to the movement of the granules towards the membrane. As a result, the vesicle fuses, all the granules releases, all the granules break down, all the chemical mediators are releasing, then they will show the type 1 hypersensitivity or the severe allergic reaction. This is one pathway, and look into the another. Same. Follow me correctly. Allergen, Fc epsilon R1 receptor. Once it is cross-linked with IgE, same. Fc epsilon R1 is going to get activated, and it is same process. There is no change. It is going to activate the protein tyrosine kinase. Here, friends, what the protein tyrosine kinase will activate? You remember right here? Here, PTK activated the phospholipase C. Correct, na? Friends, here, PTK activated the phospholipase A2. What is that? phospholipase A2. You know what this phospholipase A2 will do? It will convert the phosphatidylserine. You know, right? There are three major lipids are there: phosphatidylserine, phosphatidylethanolamine, phosphatidylcholine. Phospholipase A2 activate the PS. PS will stimulate the PE. Now you know PE in the presence of PMT1 and PMT2. You don't need to remember. Just remember that phosphatidylserine converted into ethanolamine. Ethanolamine in the presence of PMT1 and PMT2, it is converting into phosphatidylcholine. Now this is the most important. What the phosphatidylcholine will do, friends? It is going to break down arachidonic acid into what it break down the arachidonic acid, friends. It break down the arachidonic acid into leukotrienes. Then, friends, followed by the prostaglandins. What are prostaglandins, friends? We discussed that prostaglandins are the chemicals. Very important, friends. They are the one which stimulate the pain. Okay, listen my statements very correctly, everyone. Listen to me correctly. Suppose example, I am having a severe headache or I am having a severe body pains. You know, the headache or body pains, it's generally caused by what, friends? Inflammation. Then we all people commonly know that for the headache, we will take the drug named as Dolo 650, which is a paracetamol. If you're having the body pains, in addition, we will take the aceclofenac or diclofenac, which we use in our daily life. You have to remember, this prostaglandins are the one which are causing that pain. Yes. And how does this prostaglandins will cause that pain? Prostaglandins, in order to show their mechanism of activity, they specifically attaches to a receptor called as a COX receptor, which means cyclooxygenase receptors. Follow me correctly. COX receptor means cyclooxygenase receptor. So prostaglandins will release in our body. They will associate with this specific receptor. Then they will stimulate the process of inflammation. Now, friends, when we take the drug, the drug like paracetamol or aceclofenac, what they will do? They block the receptor. Simple mechanism. Once they block the receptor, what's going to happen? Obviously, prostaglandin unable to associate with the receptor, then they'll never stimulate the pain. So as a result, what happened? The pain starts reducing day by day, or very slowly it will start reducing. So in a simple word, what you have to remember is prostaglandins are the chemicals which stimulates the pain along with the leukotrienes. These two are major chemicals, and how they are getting derived from the arachidonic acid. Who will break down that arachidonic acid? Phosphatidylcholine. How this phosphatidylcholine is getting activated? Phospholipase A2. How the phospholipase A2 get activated? By the PTK. PTK activation can be stimulated by the IgE whenever it is cross-linked with the allergen. And you should not think that these two pathways are happening step by step. No, never. The two pathways will occur simultaneously, because the ultimate goal is what? Ultimate goal is releasing the chemical mediators which stimulate the process of inflammation. That's why these two pathways occurs simultaneously. So what I have explained you just now is only this. The mechanism of degranulation I have explained you. How the IgE will come and attaches here, and how IgE cross-linking with the allergen, how IgE breaking down all the granules and releasing the chemical mediators. That is what we studied. And friends, in this granules, there are two type of chemical mediators are there, friends. Actually, one we called it as generally primary chemical mediators, and second one we generally called it as secondary mediators. I want you to remember without fail. Primary mediators means whether the degranulation process is happening or not, it doesn't matter, friends. They are actually produced before the process of degranulation. That's the point you have to remember. Keep it in your mind, don't get confused, which means they are already present in the vesicle, but they are not getting breakdown. They just present in the stored form. Histamine, proteases, heparin. I want you to remember. Then what's the secondary mediators? They are not produced before the degranulation. Whenever the process of degranulation is occurring, whenever IgE cross-linked with the allergen and when the granules are ready to break, only during that particular time, friends, the secondary mediators are going to get activated. Examples are leukotrienes, prostaglandins, PAF, platelet activated factor, which are involved in the activation of the platelets. So secondary mediators are produced during degranulation, primary mediators are produced before degranulation. Keep that point in your mind. Now, don't confuse, friends. What is, and see, still we are continuing the type 1 only. Type 1 and type 2 are little, longer concepts. What is the difference between the localized anaphylaxis and what is the difference between the systemic anaphylaxis? See, anaphylaxis is a condition where the inflammation or allergic conditions occur very rapidly, or allergic conditions occur at the higher rate. What, friends? Allergic conditions will be occurring at the higher rate. Then why we are calling it as the localized anaphylaxis? Since name itself indicating, it is not specific to the whole body. This reaction extremely limited only to the specific tissue. That's why we call it as the localized anaphylaxis. An example, look into this. Allergic rhinitis, asthma, atopic dermatitis. What is allergic rhinitis? Severe running nose, correct now? And this is very specific to what? Nose only. Asthma, severe constriction in the bronchi. So it is very specific to what? Only to the bronchi. Atopic dermatitis, and it is also what, friends, with respect to the skin. Correct na? Only to the skin. So what you have to understand, in these three conditions, friends, allergic reactions are taking place. In these three conditions, hypersensitivity reactions are taking place, but they are specific to the one particular tissue only. That's what we can call the localized anaphylaxis. But if you look into the systemic, here the word systemic actually representing to the whole body. An example, friends, you might observe many times, some people, when they have a seafood, when they eat a fish or crab or prawn, you might observe entire body, within fraction of minutes to hours, entire body allergic reactions will start. Entire body, a kind of swelling, continuous itching, the entire body become completely reddish. Why? Because they're completely allergic to that, and all these chemicals are releasing in every single cell of the body, and they're causing the allergic reactions throughout the body. Some people, especially the pregnant women, they're extremely sensitive or allergic to the penicillin. So that's why before giving, that's why to the pregnant women, always they'll give the alternative drug instead of the penicillin. And you take the example venom of a bee or a snake venom. Snake venom specialty is what, friends? It will block the cardiovascular system. It will block the nervous system. Once it blocks the nervous system, what happens? The body loses control and its coordination, as a result, which leads to the one side paralysis. And later what it will block the entire cardiovascular system. What happened? The blood becomes super thicker, very thick blood become. When the blood becomes very thick, heart is unable to pump that particular thicker blood, which leads to the heart attack. Obviously the person will die. So what you have to remember, these kind of antigens are not specific to one particular tissue. They are showing their action throughout the body. So that is what we can call it as a systemic anaphylaxis. Okay, see, so far what we are studying the type 1 hypersensitivity, and I told you that type 1 hypersensitivity is mediated by the elevated level of IgE, and we understand that IgE is involved in the process of degranulation. We studied very clearly what are the mechanisms that are stimulated by the IgE to show the process of degranulation. Now the question is very clear. how can we detect the allergies? In the type 1 only, follow me correctly, don't get confused, just listen to me correctly. What are the methods of detecting allergies? In previous days, we don't have much technology, but now we are having a super technology. We can detect the specific allergens very easily with, with the accuracy of 99.99%. But just listen to me carefully. Before, friends, in order to detect the allergies, we used to perform the skin test. Just listen to me carefully. Don't confuse. Don't just listen. Okay. Suppose example, I am a person. Okay. I had played in the grass. Suppose example, I spent lot of time in the region where there is a more grass is present. Just there is a region where more grass is present. So next day, I have experienced many allergic conditions. See here, formation of, kind of inflammation as well as complete reddishness on that particular area. I really don't know why this allergies is caused. So when we visit the physician in the before days, what they used to do? First, they want to understand because of what these allergies are caused. So what they used to do, they used to apply different kinds of allergens on our skin. You understand, follow me correctly. Physicians really don't know because of what these allergens are caused. So to understand this, they use it to apply different type of allergens on our skin. See here, they have applied different type of allergens on our skin. Suppose example, if the allergy is caused by the grass, then what happens, friends? At that particular area, more amount of the mast cells get accumulated, more amount of the mast cells, which indicates that the allergy is caused by the grass. But you can ask me, sir, is it really an accurate test? No, not at all. Now, let me tell you what is the disadvantage. Suppose example, I got the allergy here, but I really don't know because of what. Now, when the doctor has applied all these allergens, remember, we understood that we have not exposure to these allergens at all. So as a result, what you have to understand, these allergens did not cause this allergy. Now, some people, what happened, since follow me correctly, in order to perform the test, we have exposed these allergens to the patient. Now, some patients, they may show the sensitivity towards these allergens. Once again, it caused the reaction. That's why it is not an accurate test. Remember my words. Initially, I did not expose to any of these allergens. Just for the test purpose, I have exposure to these allergens. What happened now? Since they are all new allergens, some people's body show the sensitivity towards this. Then as a result, what happened? New allergy reactions are causing. We don't know because of what allergy is caused. Now we are getting the new allergies. But most of the time people used to follow this particular test in the olden days because it is inexpensive and quick. But what is the point I told you? It may sensitize one to new antigens. Correct now? See, before I did not expose it to these antigens at all, but newly I have exposed. What happened in my body? It may cause late phase reactions. That's why, friends, this skin tests are not at all accurate test. Now what we are doing? In order to detect the allergies, follow me without confusion, we are using two powerful tests. One we called as RIST and one we can call RAST. You can get the question directly in a part B question or a 2-mark question. Okay, which of the following tests are the confirmatory test for the allergens, RIST and RAST? Listen my statement correctly. First point. In my body, allergen is present or not. How can I know? First point, listen my statements. I can easily confirm whether in my body allergen is present or not by the level of IgE. Why? We studied very clearly. If an allergen enter in our body, what is happening, friends? The concentration of the IgE is elevated. Simple point. Because we studied IgE is the only antibody which is releasing at a very low concentration in our body. So if there is a allergen, obviously what is happening? The level of IgE is increasing. So what is the conclusion? If the level of IgE is elevated more than the normal, which indicates that there is a presence of allergen. So we confirmed there is a allergen. How can we confirm? By the level of IgE. How can we measure the level of IgE? By using wrist. Follow me correctly. So wrist is a technique which is used not to study allergen. Okay. Wrist is a technique which is used to understand the level of IgE. That's the point I want you to remember. Now, okay, we confirmed. What we confirmed? There is a IgE level is rising more than the normal. If it is rising more than the normal, which indicates that allergen. But how can we start the treatment? If we want to start the treatment, first we have to understand because of what allergen, because of what antigen the level of IgE is increased. If we don't know what kind of allergen it is, we can't give this specific medication, right? So in order to understand what allergen specifically elevated the level of IgE, so for that reason we will do the test of RAST. Without confusion, keep it in your mind. RIST is the test which is used to understand the level of IgE, whether it is elevated or not. And RAST is the test which is used to understand because of what allergen the specific IgE level got elevated. That is the RAST test. Don't get confused between those two. Now you look into the picture. Just understand, it's a small experiment. This is anti-IgE. Name itself indicating what is anti-IgE. This is the one which will come and associated with the immunoglobulin E. Okay, that's what we can call the anti-IgE. That anti-IgE is associated with a paper disc or a agarose bead. Now, what we have done from the patient blood, what I have taken? Sorry, from the patient I have taken a blood sample. Now you know very well the blood sample contains what? Suppose example, if the person contain the allergen, obviously the level of IgE will become more in the blood. So in the patient IgE, what is there? Sorry, in the patient blood, we can observe the IgE. Now every student remember. Now these are all what? Anti-IgE. Now this anti-IgE will go and recognizes what? The anti-IgE will go and recognizes the IgE. Okay. Now what is confirmed? Now one confirmation is that the blood contain antibody. Simple. The blood contain elevated level of antibody. How can we confirm? Because whatever the anti-IgE we have taken, that anti-IgE go and associated with the patient IgE. But we know that there is a elevated level of IgE. There is no doubt. But what is the level? How much level it is? That we can measure by the radio labeled anti-IgE. So now this entire complex which will go and associated, radio labeled anti-IgE will go and associated. What is the function of this radio labeled anti-IgE? It shows the radioactivity. That radioactivity can be represented in the form of a color. So it depending on the color. An example, suppose red color, which indicates that, just example I'm giving you, high level of IgE. Suppose example, another green color, which indicates that low level of IgE is another color, which indicates that there is a no change in the IgE level. So this is what test difference RIST. In the RIST, our ultimate goal is to know whether the IgE concentration is elevated more than the normal or not. That's the ultimate goal. Very simple goal. Once again, follow me very quickly. Anti-IgE we have taken, which is associated with the agarose bead. And from the patient, we have taken the blood sample and we understood whether this anti-IgE associated with the IgE or not. If the anti-IgE associated with the IgE, which means that IgE level is fluctuated in the blood, it is not the normal condition. Now, in order to understand what is its level, how much it is fluctuated, how much it is elevated, we use the radio labeled IgE. Now, you have to understand, radio labeled IgE cannot associated with the IgE directly. That is the point. The radio labeled IgE, see here, look into this. It always associated with the complex. What is this complex? The complex of IgE, the complex of anti-IgE. To this complex, radio labeled anti-IgE will come and associated. And based on the color it given, we can determine whether the level, how much the IgE level is elevated. This is the RIST. In the RAST, what we do, friends? Instead of antibody, we will take the antigen. What we'll do, don't think that we will take only one allergen. No, we don't know, right? We really don't know because of what allergen the concentration is increased. We really don't know. So that's why what we do, we take the multiple allergens. We take the multiple allergens. Once we take the multiple allergens, then we will observe the IgE. Suppose example, I have taken 1, 2, 3, 4, 5, 6 allergens I have taken. And once we take the blood sample from the patient, then we can observe among these 6 antigens or allergens, IgE will come and at, suppose example, IgE comes and associated very specifically to the allergen 3. Then what's the conclusion? Because suppose this is the dust. Because of this, the IgE level is elevated, which means allergen 1, 2, 4, 5, 6, they do not have a direct role or indirect role in elevating the IgE. So once we conclude which allergen is increasing the level of IgE, then only we can start the perfect treatment. For that, we will do the RAST. To understand the level of IgE, we will do the RIST. Please don't get confused. Okay, so friends, what is the common method to get rid of allergy? Of course, avoidance of contact. Some people, they're very sensitive to the animal dander. So most of the people, whatever the pets that they're growing in their home, especially dog, the dog hair, we can call it animal dander. It is a powerful allergen, not in every individual, especially atopic individual. Atopic individuals are what? Who are very sensitive. Who are very sensitive to that particular animal dander, obviously it will stimulate the level of IgE. So that's why it's better to avoid the contact. If you are very sure that you will get the allergies by eating the seafood, it's necessary to avoid it. We know very well, once the IgE level is increasing, obviously it is going to stimulate the histamine. So in order, see, we know very well the histamine releases in a very low level. You know what? I can use one word. Histamine, call it as a potent chemical. Potent chemical mediator. What is potent chemical? Potent chemical means even at a lower doses also it can show the maximum activity. Those kind of chemicals we can call potent chemicals. How can we suppress this histamine, friends? Obviously by giving the antihistamine. The best example is what, friends? Levocetirizine. Hardly we will take 2 to 5 mg, that's all. But it can show the very powerful action. But it, depending on the condition, even we can go for the steroidal, corticosteroidal drugs. Right, because of the animal dander or because of several allergens, people start affecting with asthma. What's happening in the asthma? See, this is how our bronchi look like. So obviously the air will flow very freely in the bronchi. Once the histamine releases, what the histamine will do? It will constrict the bronchi completely. When it constricts the bronchi, air cannot go, right? Now, when we take a corticosteroid like salbutamol, an example, which is present in the inhaler. So, right, people will use the inhalers for asthma. Then obviously bronchi will come back to the normal position. Then the air will become free. So these are all what a type of antihistaminic drugs. In the rare condition, we will go for the therapy. Here, therapy means a combination of multiple. Combination of drugs also, as well as a combination of other treatments. That's what here indicating the therapy. So we can easily avoid these allergens by just avoiding the contact with that specific allergen. This is the best method. We can avoid it in these initial stages only, instead of taking the drugs or instead of undergoing with the therapy. Okay. This is what all about only one type of hypersensitivity, that is type 1. As I highlighted, type 1 and type 2 both are extremely important. Okay, follow me. So when we studied the type 1, we concluded a statement that type 1 is exclusively caused by the elevated level of IgE. That is what I've highlighted. When you look into the type 2, what do you have to remember? Type 2, I said that antibody mediated. Very simple point. If you quickly recall, once again, everyone just pay attention towards the diagram. Follow me correctly without confusion. Listen to me. This is an antigen, is it? This is an antigen present on the targeted cell. Now, this antigen wants to be killed by which cell? By the powerful cytotoxic T cell. Correct, na? Now, how does the cytotoxic T cell going to destruct this targeted cell? You know that very well, by releasing several perforins, granzyme, caspases. Finally, they destruct the targeted cell. Now, the question is, my friends, if the cytotoxic T cell wants to destruct this cell, first it has to recognize the cell. That's the question. And friends, how it is recognizing? Look into the picture. You can see here there is a presence of FC receptor on the cytotoxic T cell. There is a presence of antibody. Now, here I am not using the word which antibody it is. Many antibodies are involved in the cytotoxic T cell activation. You take an example of G especially. So it is cross-linked with that antigen, then it is getting activated. Then it releases the chemicals and degrade the cell. On other hand, listen, this is the targeted cell. Look into the picture, and this is the antigen. Correct, na? Now, what you can observe, this targeted cell want to destructed by the complement system. We have all studied very clearly, friends. How does the complement system is going to show their action? By the formation of a MAC complex, which we can call the membrane attack complex. Now, this is the complement system. If the complement system want to show the membrane attack complex and destruct the targeted cell, first, it has to recognize that antigen. How they can recognize the antigen? Once again, with the help of antibody. So in one line, what I want to conclude, in the type 2 hypersensitivity, if the targeted cell wants to be destructed, there should be compulsory the presence of antibody. That's the reason we will be calling it as the antibody mediated hypersensitivity. Correct, na? So remember, don't get confused. I have already told you, friends, cytotoxic T cell, if it want to show their action, it has to associate it with the targeted antigen present on the cell. So what we can remember, the cytotoxic T cell will show cell mediated cytotoxicity. What it will show? Cell mediated. When it comes to the natural killer cells, I hope you remember, natural killer cell compulsory associated with the antibody, then only they can show their activity. What do we call it this as? Antibody dependent cell mediated cytotoxicity. We can call ADCC. Not only that, friends, even complement molecule also will follow the ADCC. Don't confuse between the cell to cell mediated and antibody. Cell to cell mediated means the targeted cell associated with the cytotoxic T cell. Then only cytotoxic T cell release the chemical mediators, and then only it can destruct the targeted cell. That's what we can call cell mediated. Both are interacting with each other. But natural killer and complement molecule not associated with the targeted antigen directly. No. They are associated with the antibody. Antibody cross-linked with antigen. That's why it is called as a ADCC. So what are the examples we have to study in the type 2 hypersensitivity? All are important, friends. If not from the type 1, there are great chances are there that we can get the questions from the type 2. Hemolytic disease of the newborn, drug-induced hemolytic anemia, ABO blood group, hemolysis on transfusion, or immediate or delayed type. Anyway, this we will study in the type 4. Now remember, antibody mediated type 2 hypersensitivity. Friends, just follow me correctly. Once again, you people read in the human physiology also. Suppose we all know very well, very commonly, if the blood group is A, or suppose this is the blood group, on the antigen of the RBC, what kind of, on the surface of RBC, what kind of antigen is present? A. And you all know very well, in the blood plasma, there is a presence of anti-B. By that, anti-B never attack on this A. Suppose if the blood group is B, on the surface of RBC, there is a presence of B antigen, and in the blood plasma, there is a presence of anti-A. And you people commonly know that if the blood group is AB, is there any presence of any antigen on the surface? No. What kind? I'm sorry. The presence of antigens are A and B. And is there any antibody? There will be a no antibody, because friends, if either anti-A is present or anti-B is present, they will go and destruct these antigens. So there will be no presence of any antibodies in the serum. Now, if you look into the blood group O, what kind of antigen will be present on the surface? There will be a no antigen. And what kind of antibodies will be present in the serum? Anti-A and anti-B. This we all commonly know that. What I want you people to remember for the exam point of view, friends, you know, you have done these experiments during your first year BScs also. Small experiments. A simple confirmatory test to understand the blood groups. What is that, friends? The presence of sugars on the terminal position. Listen my words. Presence of the sugars on the terminal positions. It is also one of the major indicative method to understand the blood group. So in the O antigen, friends, on the terminal position, it contains fucose, galactose, and N-acetyl glucosamine. But mostly at the terminal positions, friends, it contains the fucose and lot of galactose derivatives. But if you look into the antigen A, see here at the terminal position, galactose is present, fucose is present. And here in the A antigen, majorly what is present? N-acetyl galactosamine is present, NAG. In sometimes instead of galactosamine, there is a presence of the glucosamine also. But glucosamine is very common in the O antigen. Galactosamine is very common in the A antigen. And if you look into the B antigen, friends, the same. Here also there is an abundant amount of the galactoses. Okay, don't confuse between the O and B. In the O antigen, galactose associated with each other by glucosamine, followed by the fucose. Here, two galactoses and there is a presence of one fucose only at the terminal position. I am not saying that this is the major and accurate method, but this is one of the indicative method to confirm what specific antigen contain what kind of sugars at their terminal positions in order to understand the blood grouping, right? Okay. So in the tomorrow class, we will study the other examples like erythroblastosis fetalis, which is very important one. Okay. By tomorrow, I'll finish this and we will go through the inflammation process. Okay, maximum in 1 or 2 days the immunology will be completed. Then we'll go to the cancer as well as the host parasite interactions. Okay. Clear everyone so far? Please revise this without fail because we'll forget it very fast. Clear everyone? If you have any doubts, you can ask. Okay, perfect. Please revise this one without fail. Okay. Okay, friends. I'll see you in the tomorrow class. Thank you all. Thank you. Thank you.